So this is really not for the non-immunologist. The first slide was basically, I gave this at the Wellness Summit that Dr. Michael Roizen has put on for over 2 decades. But I've kind of ramped this up for our purposes, knowing that you guys are ahead of the curve. But the idea of this is to give a 30,000-foot view of the integrated immune system, so that these next topics by Silverman and Rigby and Looney will resonate more. I thought I would do this as an interactive presentation. Interactive in as much as I'm going to give you a scenario, how the immune system actually works. And then we're going to take a dive in to see how its pieces and parts respond. I will touch on a few very contemporary topics. I'll just introduce you to the microbiome. Greg will give you a deep dive into this. I'm going to talk about neuropsychoimmunology because this is very important to what we'll be talking about tomorrow and beyond. And this was, again, this slide was just an introductory slide. It's not, we're 2018 here. So let's start out. Here we are, we're in Florida. Pretty cool place. And, you know, we're sitting here by the beach, and, you know, you are a human. I take that. And you have to interact in this environment here for this grueling 3 days. It's pretty tough stuff, you know. That's why we picked this. So the environment that we're sitting in is interesting. I mean, if you're looking at this from another world, you know, the planet is kind of a toxic environment. Geez, our atmosphere is kind of thinning. There's all this solar radiation, and I think I got a little bit yesterday in my walk down the beach. We have a lot of oxygen, but there's all this crap in the air. You know, global warming does occur, and the environment is threatened. So if you have an issue with that, we'll refund your money and you can leave right now. Right. And I will take no questions on that. And there's also this crazy microbial environment in our world, and it's everywhere. It's everywhere. And these humans that live on the planet are covered with microbes. They're infested with them, inside, outside. And it's not just bacteria. There's a range of prokaryotes, archaea, fungi, the microbiome. In fact, these poor humans that live on this planet, each one of them that perambulates is vastly outnumbered by the microbial species that they carry. It's kind of like, you know, you see these rhinoceri and the little birds on top of them. Well, we're just carrying around the whole microbial world with us. And like, if you're looking at that, you know, how many millennia did it take to describe microbes to begin with? And then only recently did we even start to wonder, like, what's up with that? Now we have this precious understanding of the interrelationship of our microbiome and the body. So at any rate, each human on this planet is equipped with a personal defense network. And it's interesting. And if you look at the actual phylogenetic tree, what we're seeing here is that vertebrates are over here, and at the tip of this are Homo sapiens. As we dissect the human immune system, and as all of you are aware, it's composed of innate and adaptive immunity. Adaptive immunity is only in this little tip of the Earth's phylogenetic tree, And it was late to develop and probably a product of the last about 600 million years. So interesting concept here. Just trying to get a lay of the land of what's going on. So if we wanted to drill down and look at these humans and understand this immune system, it appears to be in three highly overlapping layers. There is this kind of outer layer. The skin of humans is rather soft and quite penetrable. And they breathe and they eat. So invaders, toxins, generally reach the inner workings of these Homo sapiens by penetrating their skin, by breathing stuff, or eating stuff. That's it. And so there are special defenses located all around. We have mucociliary defenses. We have low pH in the gastrointestinal tract. The skin is enriched with a border that contains cells of adaptive immunity. Decades ago, this was considered more an integumentary defense, but now we know that it is robustly complex, and that cells of innate immunity secrete soluble products such as the wide spectrum of antimicrobial peptides that have the capability of triggering cells in innate immunity and then later calling for adaptive immunity that we'll get into. You're sophisticated, but I mean, I think it's important to note that this is not an inert layer. This is intensely biologically active. There's a few dermatologists in the office, in the room here. You know that the susceptibility of a patient with psoriatic arthritis to skin infection and the susceptibility of a patient with atopic dermatitis for skin infection are radically different, largely due to resident immune responses within the integument. The lymphoid infiltrate of psoriasis is rich with antimicrobial peptides, where the atopics are not. This is a fabulous article, and this came out in December of last year by Green and Loscalzo, and it says, "Putting the Patient Back Together, Social Medicine, Network Medicine, and the Limits of Reductionism." First of all, it's a beautiful title. It's a very eruditely written article. And it basically reminds us that our understanding of medicine in general, not talking about immunology, but medicine in general, is really a product of the past 3 or 4 centuries. Before that, before nosology of disease, you were sick, you weren't sick. You had, some kind of fire or not fire. It was just, there was no concept that this person over here with fever and diarrhea actually had a disease that the next person that had fever and diarrhea. They're both just, you're not normal, not well. Through, this rapid advance in medicine of the past 200 years, the development of the physical exam by Arenbrugger, developing the skill of percussion, and Laennec to hear with a better ear, they actually could make observations about the internal workings of a body and then take the patient down to the necropsy room and correlate this, that this suction splash that I heard was this big pyogenic abscess in the lung. This is huge. This was tectonic. Race that forward 50 years when everything moved from Paris to Berlin, and laboratory studies, Mueller and others, started to appreciate that you could actually measure parameters in the blood that reflected perturbations of normal state of health. As we turn the 20th century and the ability to see with even a more brilliant eye, with the development of electron microscopy, histology, the birth of immunology. The birth of immunology as a disease really dates back to only about 1890. The first Nobel Prize in Physiology and Medicine, as we know it today, was given to Emil von Behring. Emil von Behring had developed the first serum therapy. And, you could immunize a horse and harvest the serum and give it to a patient with diphtheria, and something good would happen. The immunologic basis of this was poorly understood. Matter of fact, it was not understood at all. Louis Pasteur, in his understanding of immunology, was virtually nil. He thought that vaccines might create something that competed for nutrients, and that's why the invader didn't come about. So, it was really Ehrlich who not only helped Emil von Behring actually quantify serotherapy, but it was his brilliant mind that postulated that there were immunocytes that had cellular receptors that could be triggered by a high degree of specificity of foreign invaders, and the field of immunology was off, particularly in serotherapy. At the same time, Metchnikoff, looking at under the microscope in Sicily, found these anemones that had these phagocytes that would respond to injury. So, our profession is relatively young. Fast forward that to today, and that's why we're all having trouble understanding this, because now, you can't really go to an immunology meeting that the major topics are on trying to divine data coming from these big data studies, where everything is understood in terms of its molecular anatomy. We'll be talking extensively about the microbiome, we talk about the proteome, we talk about the genome, the metabolome, and beyond. And so for those of us that are trying to reorient ourselves in immunology, being confronted with this molecular reductionism, we lose sight of how the whole immune system functions. And that's why we're starting out today with this kind of semi-remedial view. So I really refer you to this beautiful, beautiful Medicine and Society article. I've read it several times. And it reminds us that something that we are talking about at this meeting for the first time is that our immune response is connected to us as human beings in a biosocial model, and that everything that we do as individuals, our behavior and our choices, influence our immune system. And that is the theme of this meeting. And tomorrow we'll be dissecting this, but as we pollute ourselves with tobacco or drugs or excesses of alcohol, as we are subjected to this environment that's being degraded around us, our immune system are affected. As we live in this world, modern world that is just laden with stresses, there's an incredible literature of how stress adversely affects the immune system. And there are things that we can do to enhance it, and that is the subject of tomorrow. So that's the kind of the thing. So what triggers this immune response? This is actually only a product of the past 70 to 80 years that we really had a more granular understanding of this. Sir Frank McFarlane Burnett in the 50s had suggested that immunity was triggered by foreignness, these forbidden clones, that things that we were not used to seeing in our bodies would be triggered. And that theory moved the field ahead in a major way, won a Nobel Prize, yet at the same time was an incomplete response because many foreign antigens, all the stuff that we're eating and coming into contact with, we seem to live with relatively peacefully. The notion that infections trigger this is very appealing, right? It's appealing because infections are up until the microbial era, the reason most people died. Yet in the microbiomic world, we understand that we live in this state of immunologic détente with an incredible number of infections. So there must be something different than that. The more recent theory that has been proposed, and this has been morphed many times since this was initially described by Polly Matzinger, the danger theory, which I think holds, it's easy for us to get our heads around. That it's not the foreignness, it's not the infections, it's what is the threat to our, to these Homo sapiens on our planet. And as I will point out, those threats come from both external and internal sources. So there are a series of common phenotypic signals that tend to turn our immune system on. And we can think of them as those associated with infections and those that are not. The rubrics are, which I'm sure you're all familiar with, are pathogen-associated molecular patterns. Those are the fingerprints that are contained on pathogens that are identified by components of the innate immune system that say this is a danger to the host or it's not. They have repeating sequences. They don't use molecules that are foreign to the Homo sapien, but they are rearranged in a way or molecularly altered in a way by their glycosylation pattern or whatever that will be triggered by a series of receptors that we'll talk about in a moment. And so we call them PAMPs. More recently, in the evolution of the danger theory, is that there are also damage-associated molecular patterns that come not from infections, but can come from other sources, toxic sources, and our own internal metabolism. Every day we're turning over billions and billions and beyond of dead and dying cells. The garbage of this programmed death has to be taken out in a very orderly fashion. Proteins that are spewed into the surrounding milieu, or DNA that is not packaged in these nucleosomal packets can become highly inflammatory and thus can trigger the immune system ourselves. What happens, 5% of this population, 5% of us in this room will put out uric acid crystals. This is a classical damp. And every person in this room knows what it can do to light up the immune system. So PAMPs and DAMPs are the common parlance that turn on, ignite the immune response. Now, more recently, additional danger signals have been identified that really make this a much more complex and alluring system. And these extrinsic models trigger not through classical PAMPs and DAMPs, but this now brings in the neuroendo immunologic system. And over here on the left, we see this hypothalamic pituitary adrenal axis that we know that in conditions of stress, not acute stress, not run away from the tiger stress, but the chronic stress of trying to figure out MIPS and macra and stuff like that. This makes you secrete discharge central hormones and releasing factors like ACTH, and ultimately increase glucocorticoids. Glucocorticoids have glucocorticoid receptors on cells of innate and adaptive immunity and glucocorticoid responsive elements in their DNA. And this is an immunologic pathway. We know that the basic anatomy of the nervous system of sympathetic and parasympathetic systems are also intimately involved in tonic control of immunity. And this has been demonstrated in numerous experimental models. Again, this is the 30,000-foot view, but making the point that sympathetic discharge can have pro-inflammatory effects, and parasympathetic discharge, particularly vagal nerve, which I'll mention as we move along, can ameliorate this at a molecular level. Blunts the effects of inflammatory cytokines. We can protect preclinical models of sepsis by just modulating their nervous system. So that's interesting. So, how does that go into protecting this Homo sapien that's sitting on the planet here today? Well, it doesn't take much in this world to understand that our environment, and that means our physical environment with pollution, actinic exposure, exposure to drugs, alcohol, things and tobacco that people pollute their bodies with, and the chronic stress of modern society, all are interactive with our immune system. So it's not, the whole theme of this meeting is that, we're immunologists, we're image guys. We take care of patients with this disease, and we diagnose it, and we have these drugs over here. There are other people over here in this world that are kind of interested in stress, diet, nutrition, and over there. It's not two things. They're the same patient. As Eric Cassell says, there's mind and body. There's not mind or body. It's only one thing. So we're going to explore in a scientific way how these things go together. So that's the overview of how this defense network is triggered. These things that we'll be talking about tomorrow largely are all highly interactive with this system, and we're going to explore some of the evidence about that. So let's go back to our human subject. Sitting out here on a good day, this could be Florida, but it could be somewhere in the Caribbean, having a great time, and then this happens. So, I haven't studied it's getting, it's been the winter time, and mosquito population has gone down, but this is a Asian tiger mosquito. This is Aedes aegypti. You can see by its stripes, etc. And that can carry a lot of bad things. And you have had autochthonous cases of Zika in Florida. And well, they haven't been here recently, but let's just pretend for a bit. And this mosquito, which bites during the day, carrying this pathogen, and injects, takes a blood meal, and flushes this virus, which we'll use Zika for the moment, into this dermal-subdermal anatomic network. As I've said, all right, so now we have this defense mechanism, and here we have this components of innate immunity. I talked about the integument being important, and it is. The skin not only forms a physical barrier, but this has been breached. There are chemicals such as fatty acids that often can saponify certain toxins and invaders. But more importantly, there's this very elaborate mechanism of immune-competent molecules like defensins that can be released by cells of innate immunity that can neutralize a variety of infectious and non-infectious danger signals. So this is, the reason that this is there is to stop the invasion now, immediately. In this case, it doesn't work. And the virus has access to this Homo sapien and starts to multiply, first loco-regionally, and this requires engagement of a subsequent layer of immunity, and that is that we kind of functionally call innate immunity. And these are not separate, they're highly entwined, but it's easy to describe. And this is the definition, and I'm sure most of you can identify all of these. These are defenses without memory. They don't build up the next time you see them. You just respond each time. It is an early response, generally considered in this kind of 4 to 96 hour window. It has both soluble and cellular elements. These are germline encoded. That means that they are not the product of recombination. We have a set cassette of molecules that serve to defend us, and they recognize these PAMPs and DAMPs. PAMPs and DAMPs and the innate immune system is how this system goes. I like to think of this, and when I explain this to non-immunologists, that these are low-tech weapons. The reason that they're low-tech is they do not cause us to, they don't make us expend a lot of energy or a lot of our genome to produce them. We have soluble elements such as cytokines, antimicrobial peptides, complement, defense collagens, collectins, etc., all available as early weapons in engaging innate immunity. Yet at the same time, there are a variety of cells that subserve the innate immune system. And it's interesting to note that there's overlap between innate and adaptive, and this will become clearer as we discuss this, and as most of you know. The foot soldiers of this system are neutrophils, produced in the hundreds of billions every day with very short half-life when they're unstimulated. Basophils, eosinophils, monocytes, resident macrophages, and dendritic cells. Dendritic cells being very pivotal in linking these together. But many of these cells may also have FC receptors, which bind to immunoglobulin, which are made by adaptive immunity. So these are not just out there. These are not in airtight compartments. They are highly engaged. So how do these PAMPs and DAMPs trigger the immune system? Well, on the other side, on the Homo sapien side, with this defense network that we call the immune system, there are a series of receptors of conserved morphology that recognize PAMPs and DAMPs and can be triggered. And this system is highly complex, but there are a few that are canonical and that we have to be able to identify. Toll-like receptors now are, well known. For anything that the Nobel Prize has been awarded, you should know that because it's been canonized. And 3 Nobel Prizes were given in 2011 for work on toll-like receptors, and there are a couple handful of them. They recognize danger signals of certain types, and if you think about it teleologically, it makes a lot of sense. There are toll-like receptors located on the outside and inside the cell. There are some that respond to double-stranded DNA. Makes sense. Double-stranded RNA. Makes sense, right? Because our cellular system, we don't make double-stranded RNA, and when you see it, it tantamount that it's a virus. There are others that bind to DNA. How does that work? We have DNA. We're disposing of these trillions of cells every day. Well, when we package this, it is protected from these toll-like receptors, and that by patterns of methylation, it is further imprinted as either dangerous or non-dangerous. Pathogens are not capable of enhanced methylation, so their patterns of DNA are a danger signal. There are patterns that recognize flagellin, speaks for itself, lipopolysaccharide, and other forms of glycans. So toll-like receptors are extremely important. Now, if it were only that simple, it would be great. But there are other families of receptors. And some of the major ones are the nod-like receptors that are located largely internal to the cell that can turn on inflammatory IL-1 pathways and can bind to microbial pathogens and other forms of danger signals such as urate crystals, etc. And more recently, Rig-like receptors that were initially identified as viral receptors but clearly can recognize fungal and bacterial pathogens as well. And as everything in immunology, every time something is identified, it merely opens a door that shows you 1,000 more doors. And I think that I just wrote an editorial for a book on the human microbiome and rheumatic disease. I said we're at the same point in the microbiome and rheumatic disease as we were with the Human Genome Project 20 years ago. Let's just get that genome, let's just sequence it, and we'll know everything. And what we knew is that there's a million more questions lying ahead. So these are just a few of these receptors. Then the question is, what do these receptors do? All right, so if they can recognize PAMPs and DAMPs, there's a couple things that are important to understand. There are two basic pathways that are lit up by these, and there are more, by the way, internal. The whole of this notion of this interferon regulatory pathway and STING pathways and C-gas and others. So keep your eye on the ball for additional receptors. So some of them turn on interferon. And interferon, as we recognize, every person in here appreciates that this is very potent in our antiviral armamentarium and has a lot of other broad-reaching biologic effects. But only a few of them turn on, those that recognize viruses, very important. The other pathway that is turned on is this inflammatory pathway. And that NF-kappa B, we're now in the 25th year of NF-kappa B biology. This generates a variety of inflammatory signals and pathways, and we as rheumatologists are well familiar with this because the inflammatory cytokines that we neutralize with our drugs all wind up with NF-kB, which is intimately and intricately, rather, arranged to be tightly controlled. So when a TLR or NLR is triggered, this may become activated, generating inflammatory cytokines, the acute phase response, and all downstream effects that produce this torrent of inflammation. So what are the drivers of inflammation? Cytokines, and this is, again, you know, when I'm speaking to a non-immunology audience, this is a heavy stepping. For you guys, you get this, but, you know, the whole notion of cytokines are more than the ones that we can neutralize. There are hundreds of interleukins and chemokines, all with distinct biologic functions. Some of them tend to overlap but involve in growth, differentiation, migration. And we now recognize that while many of our drugs that target single cytokines, IL-6, TNF, they're highly effective, they don't work for everybody with these diseases, right? There's not a single anti-cytokine that just, you know, will take care of this because there's a lot of redundancy. And we now think of these diseases not diseases of single cytokines. There's a few within the primary immunodeficiency disease community. But generally, IMIDs are diseases that we should now think of as abnormalities of cascades of cytokines or networks of cytokines. This now resonates in our head a lot better, that now we can understand with greater ease why neutralizing a single cytokine does not work. You know, if we look at one of the major cells that are responsible for cytokine, and these are macrophages, and we now know that there are an infinite number of macrophage subsets by looking at mass spec cytometry, but we generally think of them as being these M1 cytokines that are highly inflammatory, and they put out all the key cytokines like IL-1, TNF, IL-6, a variety of chemokines like CXCL8 and IL-12. Each of these has jobs in terms of generating inflammation, signs and symptoms of inflammation, recruiting cells, activating cells. So this is clearly understood by you. So now we go back to our Homo sapien that's sitting on the beach, bitten by Aedes aegypti, past a Zika, could be chikungunya too, pathogen. And in fact, I think we'll flip this to chikungunya because this is where it gets better. And in 4 hours later, innate immunity is being triggered, and the ravages of that will be quite clear. And chikungunya has this bimodal phase, the first 7 days, intensely inflammatory and intensely debilitated. There have been some very nice studies that have looked at cytokine signaling patterns in this early day and gene expression patterns, and this is clearly lighting up the innate immune system. If the patient survives that first week, which they generally do, this is not a fatal disease, except for in the very old or very young, a significant number of patients now go on to develop late sequelae in terms of musculoskeletal complications that you're well familiar with. And after about 96 hours, the third part of this artificially compartmentalized defense network is engaged, and that is the adaptive immune system. The adaptive immune system is in contrast to the innate immune system. The innate immune system has the cells that we talked about. They produce these low-tech, high-volume defensive molecules to defend the host, but adaptive immunity is quite different. And this is high-tech weaponry. And if you think about it that way, it makes so much sense. Why? Because we now expend so much of our genetic stockpiles to produce these cells. You know, the PAMPs and DAMP receptors, you know, we're counting them on our fingers and toes, essentially. And there are probably many more than that. But the receptors on T cells and B cells are almost infinite, but usually described in numbers that we can't comprehend because they are not germline encoded and are produced by all the mechanisms that I think that most of you can recall. For Nobel Prizes have been awarded for that, for gene rearrangements for T cell and immunoglobulin construction. So these cells, these are lymphocytes, and they have this huge array of receptors on their surface immunoglobulins on B cells, T cell receptors on T cells, clonotypically arrayed, one cell, one receptor, tens of thousands, but clonotypically expressed. And they now need to be turned on by this pathogen that was injected under the skin, has evaded level 1 integument. The innate immune system with its resident macrophages and cells that had migrated there were incapable of ridding the danger signal. And now we want these advanced weaponry to engage. And the question is, how is this going to happen? And it happens because these antigens from the virus, this alpha virus, now can be taken up by these dendritic cells. Dendritic cells are resident. These are Langerhans cells in the skin. They have different names and different phenotypes in other organs. They're capable of binding. Their surface is literally festooned with receptors of all types, for all types of danger signals. They are complicated cells. In the resident form, they are tolerogenic, and they tend to stay in the same anatomic area. But once they bind antigen, they're not very good at killing it, and that's really not their job. They process it. This may be processed internally through endosomal processes, ultimately resurfacing on the dendritic cell. Once this processing occurs, it has determined that this is dangerous, and it receives migration orders. And now with a chemokine gradient, will migrate and enter the regional lymph node in this person. If it bit in the foot, maybe in their popliteal area or some other locoregional area. And they will set up shop in these paracortical areas where they are now looking to find a T cell that has a cognate ligand, its T cell receptor, specific for the antigen they're carrying. And they do this all the time. And if they don't find it in this lymph node, they can exit the efferent lymph and go to another lymph node. Scanning, scanning, scanning. People call this a dating service or a dance hall. It all makes a lot of sense. The job that they do at that time is really quite remarkable, and I don't think I have a slide for this, but just think about it this way. The interaction of a dendritic cell and a T cell is very impactful, because number one, it's done three things. My friend Richard Ransohoff has a beautiful slide. I thought I had it in here. He said the dendritic cell serves this danger signal up on a silver platter to the T cell there. It one, identifies it, says, is this thing dangerous or not? We have the mechanism, we know, PAMPs and DAMPs. Two, what is it and how can I kill it? And so by identifying its molecular pattern, as having viral components, having fungal components, being, some other form of prokaryote, this information is imparted in the synapse between these cells. And lastly, and remarkably, it tells this, it communicates to this adaptive cell, where can I go to kill it? If it's, you don't want all your lymphocytes going to your brain if the pathogen's in your bowel. So there are receptors that can direct through chemokine gradients to any part of the body, and going back to the skin has been probably one of the best studied organs that we know of. These naive T cells, which do not have killing potential, then in the cytokine milieu can differentiate into effector cells, and you'll hear a lot more about this. And each effector cell has its own cytokine signature that play roles in defending against different types of danger signals. Intracellular bacteria, helminths, extracellular bacteria, and others that have regulatory function or supply B cell help. You'll hear much more about it at that time. You're well familiar with immunoglobulins, and I'm not going to talk about that. So now we're into day 10, and this patient is sick. And this is an old study from chikungunya showing this very inflammatory pattern of cytokine production. And now this patient, if these cytokines become too plentiful, the patient could develop some type of SIRS-like syndrome, but fever, rash, and arthritis. And so now for this person that we're looking so coolly at on this planet, this may be critical. And if multi-organ dysfunction occurs, the patient could die. So there's immunologic control. So, you know, the best thing that happens is that upon this immunologic expansion, these T cells now one cell turns into 1 million cells in a week. If we're successful at getting rid of this pathogen, then the system is downregulated and these cells contract. This has to happen every time you get a cold, right? So there's a flow of memory here. So what happens is that over a course of a week, this one naive T cell turns into 1 million T cells. We generate cytotoxic lymphocytes, CD8s. They're tending to the battle. If it's successful, 95% or probably much more than that actually die, and a few percentage of these cells shunt off into memory. As we said, innate, it has no memory. Adaptive has this memory signal. Well, that all works well and good if we can get rid of the pathogen. But if we can't, and this occurs in chronic infections, chronic viral infections, HIV, hepatitis C, hepatitis B. This actually doesn't happen in chikungunya because there's no evidence of ongoing viral replication, but I'm just making the point here about this. Also, within a tumor, within a cancer, which is expressing neoantigens, these cells now are faced with ongoing battle. And if they are still activated and producing cytokines and robust cytotoxic activity, there will be collateral damage to this Homo sapien. They'll die from the immune response more at times more than chronic hepatitis C, where you can exist viremic for 40 years with that. But if your immune system was active, this would not occur. So there's an escape mechanism that the immune system has developed, and that's called T cell exhaustion. That is a distinct pattern of T cell biology that has its own genotypic and metabolomic signature, and they just become a little sluggish. They don't produce as much inflammatory cytokines, and they don't kill as well. And these molecules that regulate T cell exhaustion have been targeted, and we're not going to talk about this significantly today, but this is the whole notion of checkpoint inhibitors. These are now the molecules that reinvigorate exhausted T cells, and two of them are CTLA4 and PD-1, and its cognate ligands. I'm not going to discuss this. And so this is what happens. Those are brakes on the immune system. You take your foot off the brake, and bad things happen. I was just talking to Abul Abbas, and he said he was giving this talk on checkpoints, and he was in Europe, and he sat there and he says, "What happens when you take your foot off the brake in your car?" And the guy in front of him said, "Nothing." Because they're driving stick shifts, and we're driving them. Just shows you there's a little cultural sensitivity to this thing. I thought it was funny. In this patient here, control was achieved. The immune system returns to rest, and we generate a memory response. So there's a couple things that I think are important to think about, and I'm not going to talk about the microbiome. I'm going to leave that to Greg. But this checkpoint inhibitor thing of cancer, this is now huge. We've talked about it at this meeting before when there were 2 drugs. Now there are 6 drugs. The PD-1 drugs are now approved not on the basis of tumor lineage. They're tumor independent as long as you have repair defects that allow robust neoantigen formation. This isn't the panacea for cancer, but about 20% of people make long-term responses that we have not seen in the past. And there are over 50 checkpoint inhibitors in early phase clinical development, and there are trials now using them in recombinatorial fashion that we actually don't know what to expect. When they work incredibly, but there is a price to be paid for these, and that is autoimmune adverse events. Very nice article in Nature Medicine from about 6 months ago by Carl June, suggesting that autoimmunity may be the Achilles heel of cancer immunotherapy, and this has to be watched very, very carefully and studied. The CNS endocrine axis I've talked about, of how stress and other factors may trigger this. Sympathetic discharge may be pro-inflammatory, and this can be extended, throughout the host by neuroimmunologic synapses. There's very interesting work going on, Kevin Tracy and others, and this has been commercialized to help adjust the ratio between sympathetic and parasympathetic discharges, looking to see if this actually can lead to amelioration of immune-mediated inflammatory diseases. I told you that in mice, when you do this, you can actually protect them from sepsis mortality. There have been a number of papers written about this microimplantation of vagal nerve stimulation that actually have a clinical effect. There's some untoward effects of this in terms of some central nervous system dysfunction, sleep apnea, things that we're trying to get our hands on. But this is an amazing area, and I think I would keep my eye on this. I'm going to just make a couple points about the microbiome in advance of Greg's talk. That when we talk about the microbiome, we're really not just talking about bacteria and prokaryotes. We're talking about fungal elements and viral elements. We're slow to include discussions of them because our technology has not enabled us to elucidate them until very recently. But now we are starting to understand what have been referred to as inter-kingdom effects. All bacteria have phages, and in certain settings, phages and bacteria actually have a predator-prey relationship. So when you're studying, a decrease in bacterial diversity in the microbiome, the question should be, what's happening to the microvirome? Are they moving in opposite directions? And we're now starting to get really interesting data from system biology work on human phages and going beyond. Joshua Lederberg, I found out since the last time I gave this talk that he actually didn't coin this term. He popularized this term. Still a very smart guy, and we'll give him great kudos for that. But I was called out on that, and the other person was quite wrong. Quite right. These terms that you'll hear today, nonobiotic, rearing animals in sterile environments, how much have we learned from that? Here you have B27 transgene animals that have the capacity to develop spondyloarthritis and psoriaform dermatitis. It doesn't happen in germ-free environments. You have to have a microbiomic milieu. So, that's been known for a long time, but people just said it's kind of like, I look at some of these things that there's so many questions in immunology now that are just epiphanies. We've been looking at these things for decades and just kind of, yeah, that's interesting. Kind of like when Charles Janeway questioned, what is this Freud's adjuvant that we've been using for the past 40 years been doing? I think it has a fundamental role in immunobiology. And up until then, it's just, you just have to use the adjuvant because that turns on the immune system. Led to the elucidation of PAMPs and DAMPs and beyond. So sometimes it just takes a prepared mind. Greg will talk about this at a clinical level, but what we do know is that the immune system does not develop in a normal or robust way without its microbial milieu. And in animals where you can manipulate this, we know that certain species of microbes can polarize the immune system in a highly inflammatory way. These single filamentous bacteria, which are common in mice, can drive this, a Th17 response and exacerbate Th17 and IL-17 diseases. We don't have an analog of that in people. Trying to reduce diseases in man to single microbes has been very, very frustrating and probably not going to be the way to go. And hence, trying to solve our problems by eating one or two microbes is a humbling experience in terms of trying to generate evidence-based efficacy for probiotics. Yet the field is getting more interesting and more complex, and new platforms are being used. There is no doubt that the microbiome is important in host defenses because animals that are nonobiotic will die of flu, they'll die of CMV. You just put them in a cage and let them be infested with normal flora, and they survive much better. This notion, which I will close on, and I'm going to, is that there's an evolving concept, and this is one of the kind of basic questions that I have fun kind of thinking about, and I'll share with you. So if we are so dependent upon our microbiome to develop our immune system, what is the relationship between us as Homo sapiens and the microbial milieu of our surroundings, yet the entire planet? And as we affect, look at the great coral reef. All right, it's not so great anymore. This is primarily a disruption of microbial milieu and habitat. So our health, if we look at from the perspective of IMIDs, and there's no doubt that epidemiology suggests that over the past 50 years, the incidence of a variety of IMIDs, MS, inflammatory bowel disease, certain other autoimmune diseases has increased. And many people have many theories of this. This is the kind of thing that usually you need a glass of white wine and you can wave your hand for this discussion, but it's a good discussion. Is it the fact that our genome has drifted so teeny tiny in the past 1,000 years, but our diets are crazy different? Your great grandparents wouldn't recognize many of the things you put in your mouth, and is that affecting us in a way? And we're going to dive into that tomorrow. Has that affected our microbiome? Or this notion that we are all connected. We are all connected. So we are the humans with our own individual microbiomes on us and in us, but we're making behavioral choices that are affecting our food supply, our exercise, how we pollute the oceans and the air. So we are really super organisms. I think it's really, it's just something interesting. If you find this at all interesting, I have a few books which I tout out. One I've been discussing for several years, the Ed Yong, I Contain Multitudes, probably the greatest scientific, for a scientific journalist. I like it better than Dirt is Good, which is written by Jack Gilbert and Rob Knight, two of the giants of the field. But Ed Yong's book is amazingly high altitude, and he does talk about coral reefs and other species as well as man. And then lastly, tomorrow we're going to be talking about this, my new favorite book, Altered Traits by Daniel Goldman and Richard Davison, and it's about the scientific basis of meditation, and we'll talk a lot about this tomorrow, so I'm going to go from there. So if you want any more of these musings, these are my musings, please follow me on Twitter, I guarantee you I'll make you laugh, if nothing else. And I hope everybody gets Helio Rheumatology, totally free, have this fabulous editorial board. I got to throw down an editorial on Scott Gottlieb yesterday, so where else do you get a chance to really get back at the administration? At any rate, all right, so I have set the table here for the deep dive that comes ahead.